Fertility Frontiers, what is a permitted embryo in law? The hashtag for the event is HFE Act, and this event has been made possible thanks to the generous sponsorship of Merck and Fertility. The UK's fertility regulator, the Human Fertilisation and Embryology Authority, the HFEA, has been tasked by the government to advise them what changes, if any, should be made to the Human Fertilisation and Embryology Act. That's the act which governs assisted conception and embryo research in the UK. And tonight we're focusing on what is a permitted embryo in law. So in 2008, the concept of a permitted embryo, an embryo that can be legally used to establish a pregnancy during assisted conception, was introduced. In order to qualify as a permitted embryo, an embryo must have been created through the fertilization of a permitted egg. That's an egg that originates in the ovaries of a woman or girl, by a permitted sperm. So that's a sperm cell that originates in the testes of a man or a boy. In order for an embryo, egg, or sperm to qualify as permitted, there must have been no alteration of its DNA. Now, there's a narrowly defined exception to this rule that allows for the use of mitochondrial donation in treatment, which we'll come on to with the panelists later. And in order for an embryo to qualify as permitted, there must have been no cells added to it from other sources. Now, these rules have been great in one way because they've ensured that radical new reproductive technologies, although they might be legitimate and permissible in research, can't be legally used in treatment until unless a decision has been made to change the law. And that, I think, has given people confidence about what's going on and some security. Now, it's worth noting that the definition of an embryo in the Act and the scientific definition are not really aligned, and that may be something that will come up in the discussion. At this event, we're going to be exploring a whole bunch of questions, and I think first of all, really, are there currently situations in which this area of law or the guidance relating to it can be difficult to interpret, because that means it's not really working for people properly. Second, if the law is revised, should the concept of a permitted embryo remain? And if we think, yeah, it should remain, should it still be defined in the same way, or does it need a bit of attention? Third, is there a case for permitting the clinical use of in vitro derived gametes, that is, sperm or eggs that have been created or matured in the lab? Such eggs or sperm wouldn't currently qualify as permitted under the definition, nor would any embryos that were created from them. So I think Robin Lovell-Badge will perhaps be addressing some issues around that. Then the next question, is there a case for widening the approaches to mitochondrial donation that may be legally used in treatment? And we have Professor Mary Herbert thinking about that one. And what issues are raised by the possibilities of genome editing? Is there a case for permitting the use of certain genome editing tools for purposes other than conventional genome editing? And then finally, some technologies in this area could potentially be used in vivo, so that's inside the body, rather than where we normally think in vitro in the lab, in which case they would fall outside the scope of the current fertility law. So is it time to think about whether this should be addressed? And if we do address it, how will we address it? So there's lots of issues there for the panel, and probably in particular for Julian Hitchcock, the lawyer on the panel. So tonight's expert panel, I've mentioned three of them, but not the first one who'd be speaking, Professor Nick Hopwood. So we have Professor Nick Hopwood, Professor Robin Lovell-Badge, Professor Mary Herbert, and Julian Hitchcock. And Nick is Professor of History of Science and Medicine, and he's also Deputy Chair of the Reproduction Strategic Research Initiative and does all those things at the University of Cambridge.
So as Sarah indicated, permitted embryo is a recent term. This graph shows its frequency in Google Books by year from 1900. Now, we shouldn't take this too seriously, but it does suggest a UK debut in 2007 with the bill that led to the Human Fertilization and Embryology Act 2008. The apparent earlier uses are spurious. The permitted embryo came out of the longer history of regulating embryos in vitro, in glass. And I would place that in a still longer history of constructing unborn entities, as we might call them, in diverse and competing ways. Now, I say constructed because although in biological terms there have been human embryos for as long as there have been humans, and it's still true that over the centuries, different cultures have framed the contents of wombs and items that came out of wombs in various ways. The big story is how, beginning in Western Europe in the 18th century, pregnancy came to be represented in terms of developing embryos and fetuses, represented in discourse and made visible. But even in the last century, the course of the last 100 years, people have talked also of egg, ovum, baby, abortus, miscarriage, unborn child, products of conception, and many more terms. They've worked to bring some of those entities together and to keep some apart. This has mattered most in battles over abortion since the 19th century. Abortion was the first field in which the law tackled embryos and particularly fetuses, and it's still the most important. But mainly as medical interventions in reproduction multiplied after around 1960, laws got involved in constructing eggs and sperm, embryos, fetuses, and babies. Laws also helped to make distinctions among fetuses. These are once based on quickening, when a woman felt a child move inside her, and more recently on viability. Laws about human embryos in vitro have been involved in distinguishing embryos too. Now, in the rest of my time, I'm going to say a little about how this led to the notion of a permitted embryo. I'll focus on the UK because this country is the focus of our discussion and the term was invented here. And I'm going to suggest that the term came out of a set of distinctions constructed in the course of a debate, most importantly between reproduction and research, and that because permitted related to placing in a woman, the legislation should be understood also as permitting the use of various categories of non-permitted embryos for research under license from the HFEA. The prohibition on reproductive use made that research possible and acceptable. And more generally, science, of course, needs regulation if it's to thrive, not to mention be socially responsible. But there are, as we already heard from Sarah, questions about how that regulation should be revised. That's not unusual or surprising. I think as long as embryos are objects of technical and social innovation, we should expect constructions of embryos to continue to change. So I'm not going to go back very far in what I want to say about human embryos in vitro, but just to say that between the 1940s and the 1970s, researchers worked on human in vitro fertilization. That is mixing egg and sperm in a dish to achieve fertilization and early development, and eventually to implant the resulting embryo into the woman's uterus. This culminated in the birth of the first so-called test tube baby, Louise Brown, in 1978. The team who helped her mother, Leslie, do this was led by embryologists Robert Edwards and Jean Purdy from Cambridge and the Oldham gynecologist Patrick Stempter. Their work was in many ways a straightforward extension of what had gone before. And as dozens of clinics were set up, it also changed reproduction forever, especially for the millions of families now made by IVF. More people now took an interest in new kinds of human embryos as these were appearing in labs, in clinics, in newspapers and journals. there was controversy, especially about those not used for reproduction, so-called spare embryos, and also about how to make clinics safe and honest, who would have access, and low success rates. So the Conservative government set up a committee of inquiry chaired by the moral philosopher Mary Warnock. Its report in 1984 proposed the establishment for clinics and research of a licensing authority with substantial lay representation. Warnock recognized their special status as entitling human embryos to some protection, but proposed that research be permitted under license up to 14 days after fertilization. So permitted here is about permitting research. And enlightened researchers did come to see that regulation would legitimate the research and that this could be a good compromise, especially when they saw how strong the opposition initially was. In a long parliamentary battle through the second half of the 1980s, Progress, the predecessor of PET, turned the tables on the anti-abortion organizations on the other side. Progress pushed the media to focus not on fetuses, but on the very early embryos, pre-embryos as they tried to have these called, and on happy families made by assisting reproduction. In the end, Warnock's recommendations fed into the Human Fertilization and Embryology Act 1990. This set up the HFEA to license treatment, storage, and research up to 14 days. And Section 3 set out the prohibitions that you can see here, notably of cross-species transfers and nuclear transplantation. It talked about not placing in a woman, but it didn't yet use the language of permitted embryos. Now, scientists and others have held this regime up as an international model of a system that was tightly regulated, so it inspired confidence, yet unusually open to innovation. But there was some discontent and there was debate about innovations. Following the cloning of Dolly the sheep, researchers developed a distinction between reproductive and therapeutic cloning, with only the former to be banned. More generally, the reproductive versus research distinction constructed with an IVF was hardened. New ways of creating embryos produced demands for a stricter definition with respect to reproduction, or open-ended one to permit further research. Now, there were also debates about aspects of parenthood. In 2004, the UK government announced a review of the Human Fertilization and Embryology Act. In 2005, the Department of Health launched a public consultation. The following year, the government published a white paper, buried within which was a proposal to ban research on hybrids of human and animal embryos. Defending these was the focus of a successful scientific lobbying campaign. So in 2007, the Human Tissue and Embryos Draft Bill proposed to allow the creation of admixed, those hybrid human embryos. This became the Human Fertilization and Embryology Act 2008. Among the amendments to the 1990 Act, the new one revisited the Section 3 prohibitions and defined permitted embryos. So no person shall place in a woman an embryo other than a permitted embryo, or any gametes other than permitted eggs or permitted sperm. Now, I'd love to know exactly how and why the new term was invented for the 2007 bill. I don't think that research has been done by historians, but maybe someone here can shed some light. But what is a permitted embryo? Well, this is really just to expand on what Sarah said in her introduction. This is the text of the relevant section, or most of it. Permitted eggs and sperm are those produced by or extracted from women and men, or girls and boys, and in which DNA has not been altered. Similarly for embryos, to which also no cell should have been added from outside. It was allowed in prescribed circumstances, a prescribed process designed to prevent the transmission of serious mitochondrial disease. There were prohibitions also in connection with genetic material not of human origin, including the admixed human embryos. So there are prohibitions on use for reproduction that were intended, as usual, to reassure, because the Act permitted the use of non-permitted embryos in research. It came into force in 2009, and was hailed by the chair of the HFEA as providing a clear framework for practice in the 21st century. But in several areas, respected commentators have been finding this part of the Act inadequate. There's been some modest extensions since 2008, notably as anticipated in relation to mitochondrial donation in 2015. But a great deal has been happening in research recently, and there are concerns that I'm confident we're going to hear quite a bit more about very soon. So just to conclude from me, like fetuses and other unborn entities, embryos have been constructed in various and competing ways. The permitted embryo is a still fairly new kind of embryo, and it has almost paradoxically permitted a good deal of research on non-permitted embryos. But in a field of active social and technical innovation, and we can't really separate the two of those, we should expect pressure for revision, though, of course, that should proceed only after full public and parliamentary debate. A question might be to what extent that revision should and can happen within the framework that Warnock and the Acts have set up.
Now, next this evening, we have Professor Robin Lovell-Badge.
I can shed maybe a tiny bit of light on the question about where the term came up, because I was heavily involved in discussing the various amendments to the Act that led up to the changes in 2008. And I think it came from the people who write the bills in the department, well, at least the contribution from the Department of Health, where they felt that rather than having, as had been before, each type of embryo, whether it's research or non-research type of embryo, for clinical use, described separately, that they wanted to simplify things. And so it was felt that if you lump everything into either permitted or non-permitted for clinical use for transferring an embryo into a woman, then it made everything much simpler to draft the Act. And so that's where it came from. And it was run by several of us, and we generally felt that it indeed simplified things, and it was an okay way of dividing embryos into categories that made sense. Now, I still think that that's likely to be the case, because there are certainly certain types of embryos that are very unlikely anyone would want to transfer them into a woman to establish pregnancy. That would include cloned embryos, hybrid embryos, chimeras. So it's quite reasonable that these should all remain as non-permitted. So you can do research on them in the lab, but not to transfer them into a woman. And I think that's reasonable. However, science has really developed at a very fast pace, and there are other areas where we are, if not there quite yet, many of us think that it's going to be soon, where embryos that have previously been classified as non-permitted should perhaps have their designation changed to being permitted, the same with gametes. So I'm going to run through several issues that relate often to one another. I'm going to talk a little bit about in vitro-derived gametes, include along the way discussion about somatogonial stem cells, and transplantation to testes, which is something that is probably would never be covered by the HFA Act, but maybe someone needs to cover it. We've got in vitro growth and maturation of oocytes, so that's something which is not currently permitted, certainly not the in vitro growth and use of an oocyte derived after that, but even though it's working quite well in animals. Heritable genome editing in vitro and perhaps in vivo, which needs to be regulated, but we're perhaps getting to the point where these techniques might be safe enough to proceed. Not yet, absolutely not yet, but maybe. I'm not going to talk about mitochondrial DNA editing or elimination of and mitochondrial replacement. That's going to be talked about by Mary Herbert. But that just reminds me that, as mentioned, in the 2008 revision of the Act, there was regulation making power was included, which provided, if you like, a nice transition from going something that was non-permitted to something that was permitted without having to reopen the whole Act. So that's why that was done, at least in part. So it's possible that some of the things we're going to talk about today could perhaps, you could change the Act to make some of these things have a regulation-making power to allow a simpler change to the Act rather than having to debate the whole thing. So we should think about that in our discussions. And I'm going to touch on, I don't want to spend much time talking about it, the 14-day rule, because this is relevant in many cases to testing the safety of some of the methods that I'm going to be talking about. And I'm going to end on stem cell, or maybe end on roughly on stem cell derived embryo models, which is something that the Act doesn't cover, but maybe it should. So maybe we're going to include another non-permitted embryo class into the Act. So let's start. We'll start with in vitro derived gametes. So we have a number of potential sources of these. So and people have been working on all of this. So you can start off in embryonic stem cell lines derived from early embryos. Of course, they would be characteristic of the embryo, not of an individual, so they're not so useful. They could be created by somatic cell nuclear transfer. You could begin with patient-specific induced pluripotent stem cells or iPS cells. So that's derived from reprogramming from adult somatic cells. We could have somatogonial stem cells from individuals who could be infertile because there's a problem with some aspect of their gonads or whatever, but these are present in testes from birth. They can be transplanted to testes if they're grown in culture. Primordial follicles from fetal gonads or early postnatal ovaries can be grown and matured in vitro, at least in animal systems, and the methods are getting there for humans. You can have direct reprogramming of primordial germ cells or gamete precursors, even from somatic cells, which is, I think, further off in terms of having a safe alternative for doing something, but this is possible. Okay, so how about somatogonial stem cells? The testis contains a special stem cell type that's able to self-renew and give rise to cells that give rise to sperm. Way back in 1994, Ralph Bristows developed methods of culturing these for extended periods of time and to transplant them back into the testes. That works best if you've depleted the testes of endogenous germ cells, and you can use simple drug treatment to do that. And they were able to show this works in mice. It's been done in rats. It's been done in non-human primates. So, and it's getting closer and closer in humans, but at the moment, there's no particularly reliable way to culture the somatogonial stem cells from human testes. But any day now, I suspect that problem is going to be solved. For the ovary and oocytes, you have other issues, but again, you can go successfully. This has been done in animal models. So you can take primordial germ cells through early meiosis, follow protocols developed by a number of groups for the growth and maturation of oocytes. For example, using 3D cultures. Some of these work very well. Occasionally you can get spontaneous differentiation. It seems like all the way from primordial germ cells to oocytes, but that's not very reliable. You can begin with pluripotent stem cells, derive primordial germ cell-like cells, combine these with somatic cells from fetal ovaries to get aggregation culture, and then transplant those to newborn ovaries. Or you can go from pluripotent stem cells, derive your PGT-like cells, and then combine those with somatic cells from fetal ovaries with aggregates that you can then maintain in 3D structures in vitro and get oocytes which are fertilizable, at least in the mouse. More recently, it's been possible not to use directly fetal ovary cells, but to derive fetal gonadal cell types or gonadal cell types from pluripotent stem cells. And so you can co-culture your primordial germ satellite cells and your gonadal cells in culture, both derived from an IPS cell, for example, and derive oocytes from these. And like I say, in the mouse, that's been shown recently to be functional. So the progress on this has been quite rapid, and it's including the mouse, but also more recently non-human primates. And it's suspected that while not yet, the progress will go very rapidly, and we will probably have human in vitro derived gametes not too far away. Let's skip that. So the most successful methods involve either transplantation of germ cell-like cells back into the testes or ovaries, or aggregation and co-culture with somatic cells from the ovaries or testes. And just mentioned, those gonadal cells can be obtained by also from pluripotent stem cells. There's several papers describing that, and which is getting better and better. This is a horrible slide to look at, but just to illustrate some of the progress being made in mouse and in human. Mouse is there, and you can get all the way to live offspring derived from these in vitro gametes. Human, we're not there, definitely not there yet, but it's getting closer and closer. Also, non-human primates definitely is getting closer and closer. So this is blastocyst development from after fertilization with in vitro spermatids derived from embryonic stem cells. So if it works in non-human primates, it's not far from working with humans. Now, I want to talk briefly about human heritable genome editing in humans. Now, just to stress at the beginning that the methods are really not safe enough yet. However, you never know what's around the corner again. There are two possible methods for doing this. You've got editing the fertilized egg or zygote, and this in some ways is challenging because you don't have a reliable way of verifying the edits you have. You would have to know that your methods are 100% or almost 100% reliable. So because of the risks of mosaicism, you can't rely on, for example, pre-implantation genetic testing. But the other route, which is essentially using in vitro derived gametes, could be safe and reliable because you can grow your genome edited cells large amounts of, you can test very carefully for whether you've got the correct edit and no another edit, and then use those to derive germ cells from which you then make an embryo for transfer. So that may be a better route. Where are we? It's often argued that we have other ways of allowing patients to have children without carrying a genetic disease, but they don't apply in all cases. So you can use pre-implantation genetic testing, for example, but they're unacceptable for some women. These always put a burden on women who have to undergo IVF. There are cases where they cannot be used to retain a parental genetic connection, but avoid having a child with a genetic disease. That would be in situations where you have an individual who is homozygous for a dominant mutation, both parents homozygous, or both potential parents homozygous for recessive mutations, et cetera, et cetera. So that's a bit of what I was saying here. There are also situations where pre-implantation genetic testing is incredibly inefficient. Savior siblings would be an example there, or where a mutation affects fertility. So you have far too few oocytes or embryos produced. So there are essentially two types of embryo model, and this was a designation from the International Society for Stem Cell Research and an update to their guidelines published last year. So you can have non-integrated stem cell-based models, such as gastroloids, which mimic part of supposedly part of normal embryo development, like the posterior end of the embryo. These are very useful for studying aspects of embryo development, but it's very clear that these could never form a normal embryo ever, because they're missing too many components. But you also can have so-called integrated stem cell-based embryo models, or blastoids, which mimic at early stages quite closely normal human embryos. And this is one example from Austin Smith's lab, but there are many others. And so these can be derived from embryonic stem cells or from iPS cells. You can get something that looks like an embryo that could perhaps be transferred. I don't know anyone who's ever tried, and maybe it's going to be done in animal models, but obviously the feeling is it probably should not be tried in humans. So this would be an argument for having a non-permitted embryo. But these aren't regulated by the HFEA, which is why I'm raising this, because first of all, if you begin with they don't cover anything with an embryonic stem cell line once it's derived. That's not in their remit, and they don't do anything about iPS cells, which are certainly not in their remit at all. So these types of things are not currently regulated or regulated completely differently, if at all. And I just want to touch on the 14-day rule because this is really important. If you want to test, for example, your in vitro derived gametes or your genome editing techniques or your MRT techniques, then it's much more informative if you can take your embryo beyond gastrulation, where you now have all the different cell layers being formed. So, so I'm not going to touch on this at all. It's really hard to do this in any other way than culturing embryos beyond 14 days. And just to point out, in non-human primates, they've been taken to the equivalent of about, I guess, about 22 days in humans. So that they can go maybe that far, at least at the moment. And this is already far enough, though, to give you a huge amount of information. So just to stress that this is one part of the Act that I personally think ought to be changed.
So I'll move on now to this evening's next speaker, Professor Mary Herbert.
So the mitochondria are known as powerhouses of the cell. They generate vast majority of the energy requirements of the cell. They contain their own genome. That's what's important here. And so if there are pathogenic variants in the mitochondrial DNA, it can cause a broad spectrum of multi-system diseases, and the prevalence is estimated to be 1 in 5,000. So that should be 1 in 5,000 of the population. And disease onset can occur in childhood or later life, and in the worst cases, children die in early childhood. And importantly, there are no curative treatments. So the mitochondrial DNA are maternally inherited. So that means that the mitochondrial DNA present in the fertilized egg comes exclusively from the egg. So the nuclear DNA, which is contained here in the pronuclei, comes from the egg, the mother, or from the sperm, the father. The mitochondrial DNA, of which human eggs are estimated to contain about 500,000 copies, comes entirely from the mother. So the pathogenic variants can be present in all copies of mitochondrial DNA or in only some. And the vast majority of known pathogenic variants coexist with normal mitochondrial DNA. And this is known as heteroplasmy. And in these cases, the severity of disease symptoms is largely dependent on the variant load. So if you have more mutation-carrying mitochondria, you're likely to have more severe disease. Now, there are some pathogenic variants that exist as homoplasmies. That is, all copies of mitochondrial DNA are affected. And in these cases, the severity of disease symptoms can vary. So a woman who has herself only very mild symptoms can have a child who's severely affected. So there's been some focus on assisted reproductive technologies to reduce the risk of transmitting mitochondrial disease. And so in most cases, pre-implantation genetic testing can be used to reduce the risk. So in this situation, a woman undergoes IVF treatment, has multiple embryos produced, and then we would take a cell from the 8-cell embryo, most usually, and send it to the diagnostic lab, and that enables us to identify the embryo with embryos with the lowest variant loads for replacement. Now, of course, this relies on the woman producing embryos with low variant loads, and this isn't always the case. It's certainly not the case for homoplasmic mutations, but in some cases of heteroplasmy as well, when there are very high levels of heteroplasmy, it's very difficult to get embryos with low enough mutation loads. So the alternative then was to develop mitochondrial replacement to reduce the risk of mitochondrial DNA disease of transmission. So the principle here is that we would just take the nuclear genome out, rescue it from its defective mitochondrial environment, and replace it into an egg donated by a woman who doesn't carry a mutation. So it involves microsurgical transplantation of the nuclear genome, and this can be performed before the egg is fertilized or after the egg is fertilized. And so I'm just going to go through, just bear with me, I just need to explain a bit of biology here. So oocyte maturation means the process by which an egg begins to prepare to be fertilized. So you've got an immature egg, and then we get the trigger to ovulate, or in IVF, we give artificial triggers for this. And then the egg begins to undergo maturation. It undergoes the first meiotic division here to lose half of its chromosomes. And then it arrests here, waiting to be fertilized. And when the sperm fertilizes it, it loses another half of its chromosome. So it ends up with just a single chromosome to transmit to the egg. So the egg then has a single copy of each chromosome from the sperm and from the egg. And then the process, fertilization is more of a process. The sperm comes in, it triggers this final division of the egg. Then we get what we call the pronuclei. They come closer together, and then you get the first mitotic division, and then we get the 2-cell embryo. And it's at the 2-cell stage, that's the first time that the parental genomes come together. Up to that stage, they've been in separate compartments. So the currently permitted stages for genome transplantation are the mature egg, the metaphase 2 egg, and there we can perform spindle transfer. The spindle is removed. And the rationale for that is because when we do stimulation for ovarian stimulation for IVF, the majority of eggs are at this stage. Legally, we can also perform nuclear genome transplantation after the egg is fertilized, when the pronuclei appear. And the reason that these were selected was because most of the research that had been performed had focused on these stages. So that's spindle transfer and pronuclear transfer. And since then, and indeed even before the law was changed at that time, in 2015, we had other possibilities coming online from research done by various groups around the world. So one possibility first published by a Chinese group was to use the polar body. So when the egg undergoes its division here to lose half of its chromosomes before fertilization, it loses half of them in a small structure called the polar body. And so there was a possibility to rescue that in order to create 2 maternal genomes per egg. And obviously there are obvious advantages from that, because we wouldn't need to do as many egg collections to get the eggs we need to do mitochondrial replacement. Another possibility that was published subsequent to the changes in the law was removal of the maternal genome shortly after fertilization. So when it's the sperm comes in, it triggers the final division, and then the egg stays in a sort of suspended animation for a little while there. And the maternal genome can be removed at that stage. And that's a really sort of conservative approach to it, because it means we don't have to transplant the sperm. So these are just other options that have been published since, and may have advantages. I think still need to be tested, but I do think that they are worthy of consideration. So another possibility is, another problem with mitochondrial replacement is that when we do the nuclear genome transplantation, we get some carryover of mitochondrial DNA. So here we have patient, I'm just showing you for purposes of demonstration pronuclear transfer. So we remove the pronuclei and there are some mitochondria contained in this, what we call the karyoplast, which contains the pronuclei and the mitochondria. And that means that when we fuse it with the enucleated patient egg, that there are some maternal mitochondria persisting. And in some cases, our research and others showed that in some cases, this tiny fraction of mitochondrial DNA can take over when we made ES cells. So there was what we call reversion to the maternal mitochondrial genome. Now, we don't know if that can happen in vivo yet, but this reversion means, or this carryover means that there can be persistence of mutated mitochondrial DNA, and this could give rise to higher mutation loads in certain tissues. So we would have to conclude from that mitochondrial donation may not prevent disease in all cases. So there's been a focus on how can we just reduce the risk of this? How can we get rid of this fraction of mitochondrial DNA that we carry over? And two possibilities are we go for selective reduction of the maternal mitochondria. So you just get rid of the mitochondria, or you can just focus on selectively reducing the mitochondrial DNA. So selective degradation of maternal mitochondria. So cells degrade mitochondria by a process known as mitophagy, and that's a machinery that cells have to get rid of defective mitochondria. But we can artificially engineer proteins to bind to the mitochondria that will then target them for mitophagy. And that is one possibility for getting rid of maternal mitochondria. And then another way is to go for selective degradation of the mitochondrial DNA. And this can be achieved. So the mitochondrial DNA is a circular molecule. If it becomes linearized, if we cut it to linearize it, then it's very rapidly degraded. And this can be achieved using nucleases. And so the idea is to target nucleases to the mitochondria so that they can access the mitochondrial DNA, cut it, and then that's very rapidly degraded. And so research is underway for both of these procedures. The mitophagy has been, I think there was a paper published quite recently on that from a Chinese group, and it was effective in reducing mitochondrial DNA carryover. This linearizing of the mitochondrial DNA is something we're working on, and it seems to be highly effective, but would still need to be tested for safety and efficacy. So just to summarize, in order to optimize in order to improve mitochondrial donation procedures and continue to optimize them, it would be good to have more possibilities to make provision for performing the nuclear genome transplantation at different stages, or in the egg, before fertilization, after fertilization, and to have the possibility to introduce adjunct technologies to reduce carryover and persistence of maternal mitochondrial DNA.
So our final speaker this evening is Julian Hitchcock. Julian is a solicitor and he's of counsel in life science regulation at Bristows. And he was a contributor to the Nuffield Council on bioethics reports, genome editing and human reproduction, and human embryo culture. And Julian is an expert on the regulation of reproductive, regenerative, and genomic medicine and research.
I want to look at these three questions before you. What are permitted embryos? Plainly, we've talked about a lot. Who permits them? And how does the law keep up? An overview, and we're looking at three basic things. First, the definitions surrounding the term. Second, the legislative process and its relationship to the technical process. And third, how fertility legislation might adapt to change. So it's difficult. Legislating for changing science and technology is fraught. Its laws quickly become embalmed with legal meaning, as we heard earlier, drifting away from scientific meaning. However, all we have to work with are words. So here's the prohibition we looked at previously. And let's just look at that first one. We'll come back to B there. But no person shall place in a woman an embryo other than a permitted embryo. Robin, of course, raised the issue of the definition of the embryo. It's certainly something I'd be very happy to speak about later. I suspect for now we're looking at the question about the permission of an embryo, but I share those views about the definition. And I would merely note at this stage that it is splendidly circular, but we can perhaps discuss that later. Here is the definition that we came across, and the piece that I put in bold, you've got the 3 criteria. I'm largely going to just put C aside, but the 3 key criteria, the one that stands out, of course, if you like, from a sort of tabloid point of view, is this one, that if you want to edit an embryo, well, you can't, because you can't. But as it's preferable, as we heard from Robin, to edit upstream of there, it probably won't arise. So let's look at another aspect of that definition. It's got to be created by the fertilization of permitted egg and permitted sperm. There we are again. So when you see that, you realize what on earth are these things? What are these permitted gametes then? Move to that. And again, we've seen this expression before in relation to the embryo, but whose nuclear or mitochondrial DNA has not been altered. And this looks like a roadblock, because isn't that exactly what you're doing? But of course, as we heard, what you actually want to be editing is the precursors, the spermatid, spermacytes, spermatogonia, and so on. So begs a really rather obvious question. This is certainly a case where you have a departure between law and scientific terms, which is what do egg and sperm mean? And the Act provides a definition here. And all the discussion we're having, I think, largely here is derived from amendments made in 2008. But what we have is this curious, and some might say slightly biblical definition, which reads the same, much the same for eggs. It sort of gets to the Delphic heart of the matter, which is references to sperm are to live human sperm, including cells of the male germline at any stage of maturity. I'm going to quickly explore that. I'll just simply remark that, again, it's circular, it's non-exclusive, it doesn't specifically refer to germ cells, and the word line is let's say, interesting. But I do want to come to the question of artificial gametes and progenitors. I used the IPS, but I appreciate that you can use ES-derived ones and other approaches. But taking that particular example, they seem to fall within the definition because, notionally at least, they are carriers of a germline. And so, in that sense, you have something which potentially falls within there. But here we come to this point, again raised earlier, produced or extracted from the ovaries of women, or if it were, the testes of a man, or if it were ovaries. And this does raise an interesting issue, because logically, sperm produced ex vivo, but grafted into testes would be permitted, whether or not the testes were attached to a man or even a living one. But of course, the question that we're facing with all these words getting out of kilter is exactly the one that Dr. Johnson, right at the top, was observing in preface to his dictionary, which is things change, attitudes, and especially when you have scientific developments taking place. So we shouldn't be surprised at this. But what we should note, of course, is that back in 2008, although we might sort of question the definitions which we have now, Parliament was indeed thinking ahead about the future. And it had this for the present purposes, and picking up on what Mary was talking about, this particular legislative purpose to prevent the transmission of serious mitochondrial disease. That's important because it's a legislative purpose, and you'll see why in a minute. So what does that mean? Well, it means that way back in 2007 and 8, earlier actually, Parliament reflected on the possibility of preventing transmission of a class of serious inherited diseases. And of course, it sees that, as you do, because we can't see the future, through the prism of this promising field of mitochondrial donation. And it legislates for the possible use. So that's what it aims to do. That's taken from the parliamentary note, pretty much. And there we are. So how does it achieve that purpose? Well, it does this. It says we can make, regulations may provide. We, Parliament, won't make those regulations. Regulations are secondary matters. But those regulations may provide that an egg becomes a permitted egg, a sperm becomes a permitted sperm, and indeed here particularly, an embryo can become a permitted embryo, even though it has applied to it a prescribed process. So what it's doing is extending the definition of a permitted embryo and indeed permitted eggs. And that's done by the Secretary of State. And of course, it's the other bit to note there is it's about this prescribed process. It's talking about something that's ultimately technical. And there it is. This is what then happens. So the regulation, only one so far that's been made in relation to that provision under 37A5, what it's worth, is the 2015 regulations, the mitochondrial donation regulations. Now, I think it'd be useful to try and explore this a little bit, the processes involved. So first of all, the technical processes. So we've heard about the two prescribed processes, but what I'd like to do is to see how we can flex the rules to add new processes to increase our ability to prevent transmission of serious disease. Now, I must say that these are hypothetical, and I'm not suggesting that there is one, but there might be. We just want to kick the tires a bit on the structure of the regulation. So prescribed processes, two of them as we've heard. And this is basically how the regulations made under the Act set them out. You've got two steps. First of all, I think we've had that described to us. And in fact, the most important word on that page is the word or, because there are only two techniques which are prescribed processes. But what I'd like to do is to suppose that there were, in fact, a better process to prevent this transmission of serious mitochondrial disease, again, in Parliament's purpose. So what if we have another technique for doing that? And so I have this notional trip called the candlelit path, which takes us down the sort of the notional steps in a development of the law. And the first candle is what essentially inferred from what we talked about, that mitochondrial genome editing in that case becomes a prescribed process. In other words, nuclear from the nucleus becomes a prescribed process. Sorry, no, I didn't say that. I just said mitochondrial genome editing becomes a new prescribed process. You'd have to change the title, of course, for the regulations because it's called donation, but I'm sure we could fix that sort of thing. And it would be easier because you wouldn't have the three-parent thing. You can see the advantages. Let's say it's a thing and it's real. And once you've done that, if you accept that, then we might say, well, what about all the mitochondrial genes that are in the nucleus? So it's consistent with the same legislative purpose that we could, for the purpose of preventing the transmission of serious mitochondrial disease, that we should be able to edit those. Why not? So the second idea would be you actually amend the Act to a higher level now, which would permit the editing of nuclear DNA for the same statutory purpose. And then we come to the third candle, which is sort of, again, the sort of logical endpoint of this candlelit journey, which is you amend the Act in a really rather simple way, which is to permit the editing of nuclear DNA to prevent heritable transmission of serious disease. You lose mitochondrial, and because you could have meant something infectious, you put inheritable. That's just a suggestion. It's just a rough idea of what could be done to extend the purpose. So that's key, because that's Parliament deciding, if it does, to extend the purpose. Now, yes, and again, I'd suggest that we use that within the same framework. So a key feature of one of the biggest problems we face from a legislative point of view is simply the very pace of change. And this actually was something recognised by the WHO committee in its report, in its recommendations to the UN. So how does legislation keep up? And the change problem could be put this way. To be legitimate democratically, the law has got to reflect social attitudes and respond expeditiously to changes in science and technology. And this is sort of how we think it's working on the face of things. You've got the Act, you've got regulations made under the Act, and there you are with the procedure. So you've got the Parliament, and then you've got the detailed stuff on the right, which is what is done by the Secretary of State. Parliament doesn't need to bother, but unfortunately that isn't true. So what actually happens here is, if you wanted to add in that new technique that we hypothesized about, actually you'd have to go into Parliament. So you'd actually, there is a provision on Section 45, which means that you can't approve the regulation unless it's been through Parliament. And that, of course, is exactly what happened with the mitochondrial regulations themselves. So it's not really a delegated power. And it means that Parliament is responsible for approving prescribed processes. Seems odd. And so the problem that we have is exacerbated by technological change. It doesn't necessarily engender trust. And I would suggest that it's not actually a model to continue. It's something to really be very wary about, and to distinguish those two roles of the legislative purpose and the technical implementation. So what do we do about it all? Well, the most obvious way is to have a dedicated technology approval body, which would participate in the international expert groups on the identification and development of international standards, such as those proposed by the International Commission and of course WHO, and the proposed Frontier Technology Interagency, amongst others. So that might be one approach, but that technology approval body would then develop and update national technical requirements and guidelines and approved technologies in the sort of way which we would expect, and who recommended open, transparent, honest, and accountable. I would also suggest that that's a role distinct from the licensing authority, but we perhaps could look at that elsewhere at another occasion. There is finally, I think, the question of what is a serious disease. Who decides what that is? It's all very well saying that Parliament much invents a notional, we have a notional new service to prevent heritable transmission of serious disease. But who's really going to decide what they are? I would suggest that that is actually ultimately a technical matter, and that we have, and if we take the example, for example, of the science advisor, if you look at the HRA's approach to PGTM, listing those, and I think it's possible to do it in such and such a way, but it should, I believe, they should be conducted, if you like, with recourse to international resource. I don't think we need to involve Parliament in the specifics. And looking at the two sort of ish candidates from the HFEA, statutory approvals committee, I don't think that's right. So currently that's involved with licensing under the regulations, but it's not a technical assessment body. It's publicly invisible, really. And by the HFEA's own internal review, is under resource. That's from 2018. So I think there's a recognition there that it's difficult. Then, of course, there's the SCAC committee, Scientific and Clinical Advances Advisory Committee. Could that do it? Well, sort of, but not, because whereas it has the scientific brief, its remit is actually too general, I would suggest. It has negligible resources. It is almost invisible publicly. It's not independent of the licensing authority, of course, and it actually has less power, literally less power, to approve new technologies to alleviate serious mitochondrial disease than an assembly of earls and bishops. That's literally the case.
Now, I'm going to go to the audience questions, and I'll start with the most popular one, which is for Robin, from somebody who's an anonymous attendee, saying, if you were allowed to culture human embryos in a dish beyond 14 days, e.g. to test genome editing techniques, how could you validate those results to be sure that what you see in the dish is close enough to what would happen in the womb? So I'll go to you, Robin, first, and then if others want to comment on that, Will.
I mean, that's an issue, of course, but what alternative do we have? So it is, of course, possible to obtain early embryos, fairly early embryos from terminations, but before about 28 days, it's really, really hard. Shankar Srinivasan in Oxford published a very nice paper in Nature towards the end of last year, where he was able to obtain a single embryo that they thought corresponded to something like a 16 to 18 day embryo. They couldn't stage it more accurately than that, because probably the way we have of staging human embryos itself is a little imprecise. But you can, if you can culture human embryos in vitro from pre-implantation stages through beyond 14 days, you could compare them in detail with embryos isolated directly from terminations in a detailed way, as was done by Shankar Srinivasan in his lab. And then at least you have some indication that what you're getting in your in vitro culture is the same as would happen in vivo. And you only need to do that a few times to be fairly sure that what you're getting in culture is appropriate. And then what you're looking for is differences once you have done your manipulation, whether it's genome editing, whether you've derived the embryo that you're going to culture for extended periods by a method like mitochondrial replacement, or after genome editing, or using in vitro derived gametes. So you have a way of verifying everything against each other if you do it that way. So I think it's possible, but there's no other way of doing it. So if you can't culture embryos beyond 14 days, the only alternative is to get real embryos that have implanted. And I think everyone would agree that that's perhaps at this stage a little too ambitious to expect things to be perfect and safe.
Mary, did you want to come in on that one at all?
I mean, I think it's important to note that this would be done for research purposes, not for treatment purposes. And I imagine the validation would be done on sort of different organ systems based on what we know already. And as Robin said, from these very valuable contributions from early embryos developing in vivo. But it is important to know this is for research, not for treatment.
I mean, for Mary, for your for the MRT, I mean, you had to take a big jump going from limited embryo culture in pre-implantation stages, and then some work on deriving embryonic stem cells from those embryos. Don't you think it would have been better if you could have gone further?
I think so, but I think even more now, if we can just increase the repertoire of tools and assays that we have to look at the safety and to look at the effect of our interventions. And I think that this is really a direction that research should be taking, not just like in the pre-implantation stages as well, so that we can look directly, because there isn't so much to go on when we're doing this type of work.
And is this then, Mary, something where the law and the restrictions that Julian, and you and then Julian outlined is, holding people, potentially going to hold people back?
So I think the sort of development of new assays and ways of assessing our interventions would be done sort of under a research license. And I think anything we need to do on that front, we can do at the moment, I think, except for culturing beyond the 14 days, but within the pre-implantation stages. For example, if we wanted to integrate a fluorescent protein that marks the chromosome so that we can look directly at the effect of the intervention on the chromosomes. And this is work we're doing in collaboration with Kathy as part of the Wellcome-funded Human Developmental Biology Initiative. I think if we can get to that stage and directly look into the embryo to see what was the effect of our intervention, that would be so much, really facilitate the introduction of new techniques and also just revisit the ones we're using.
The next question is from an anonymous attendee, which says, does the law specifically state that no embryos can be placed in the scare quotes, woman unless, if the law does specifically state woman, would this need to be amended as gender norms evolve? And I think this is something that I'll go to you with first, Julian, because when you read it now through today's lens, it does somewhat jar perhaps to some people.
Yes, it does, and it's certainly it's a thing which was put to me, and I remember feeling slightly boggled when it was first put to me that the law, section 3 says no person shall place in a woman, blah, blah, blah. And the point was made to me that actually it was possible to have a womb implant, and this was entirely feasible. It's true, you know. I mean, there's a difference between what may be technically possible and what may be legally possible, and perhaps there is also a question here about parliamentary time. I think it's a case that should be made, and I think I know who may have raised this point, but you can make that point. I have to say it's something which feels slightly tangential. It's an issue, and it's not the only one. There are other issues. We've talked about permitted embryos. Other people will have feelings about prioritizing certain embryos, for example, which doesn't technically go under the permitted embryo tag. But yes, it's certainly something which is up for discussion, no doubt.
And Nick, I was going to bring you in on this point because you know, I know that you always look closely at the language, and you know, and you discussed in your presentation, you know, how these things change over time and how society's norms change over time. So I wondered if you wanted to chip in on this.
Well, it's certainly something that leapt out at me when I was rereading the Act earlier today, and certainly it's. I mean, to me, it's hard to imagine that shouldn't be revised so as to include trans men, for example, and maybe especially. Yeah, I mean, historians are used to reading documents, sometimes even from the relatively recent past, and seeing just how mores and norms have changed, and so that did really jar for me. Looking through it yesterday and today.
Thanks, Nick. Do Robin or Mary, are you wanting to come in on this one, or shall I move on to the next question?
I think we'll just agree that it's something that needs to be clearly looked at properly. I mean, a simple way is not to actually mention women, but just mention transfer into female reproductive tract, wherever, you know, oviduct or uterus, and that would be the simple way of getting over that. But it absolutely needs to be addressed.
Yes, I think it's yeah, there are. It's not beyond the wit of man or woman to see how you could perhaps do some simple changing here to the language in the Act, and not create too many problems, one would hope. The next question is from Ghulam Bahadur, who, and I think this is a question for Julian in the first instance, an embryo created without fertilization, cloning technique, is it legally an embryo or not? And if not, how did the HFEA win that legal case?
If we're talking about going back to the Quinta-Farley case, and which essentially was one of the factors which triggered the redefinition in the 2008 Act, the answer to the question is, how did Parliament come to a conclusion, is this. First of all, the problem faced by the court at that time was that Dolly, not incidentally a human, was not the product of fertilization. And at the time, under the existing the definition at the time, that was what an embryo was. How quaint. And so the House of Lords, as it was at the time, now the Supreme Court, I suppose we'd have to say, determined that actually you had to look back to what Parliament would have meant if only it had known. And it's actually not a bad approach. It sort of helps us limp along, but it's not really a substitute for parliamentary engagement and ultimately sort of consultation you've spoken about, and actually setting some good new regulatory framework. Was that the question answered, or was there something? How did they do it? Why is it illegal? I mean, the definition is the definition of an embryo is again something which we could turn to. It is gloriously circular, as is the definition of a sperm. A live human embryo. Embryo means a live human embryo. Okay, and there is an issue over that. I think that is something which does need looking at. There are a number of definitions which I think deserve merit. And so, for example, we had the ISSCR guidelines which explore that really rather well, I think, in the glossary. But actually, even in English law, as it is, rather surprisingly, but not actually under the HFE Act, it's under the Biotechnology Directive and patent law. But we do have a definition, which is, if you like, what's the word? It's entelechy driven. It's actually driven by the capacity of something to actually reach a particular state. And so that this court in the international stem cell case observed that the mere fact that the organism commences a process of development is not sufficient for it to be regarded as a human embryo. Quite a useful proposition. And it may be the sort of thing which would feed into a reconsideration of that definition.
Nick, do you want to come in now on this one?
Well, just to be clear, Julian, I mean, when you say circular, I take it that saying live human is qualifying the kind of embryo that's intended, and that you wouldn't dispute that. And you're just, if I understand correctly, you're just saying that, then saying, other than that, it's saying an embryo is an embryo.
Yes.
But the live human is clearly quite an important qualification.
Yes, it is. It is. And indeed, this was something which, of course, came up, I think Robin referred to a paper recently when actually the isolation of a post 14-day embryo and actually using that, so the independent of the Carnegie scale, actually doing an amazing analysis on that. And I was approached by someone saying, "This is legal? It's after 14 days." And the answer is, it is. It's not a live human embryo. So that part's clearly important. You have the same criterion, obviously, in relation to sperm and eggs. But it is circular. I have some sympathy with it. But if I were looking for a solution to that, I think I'd look for an ethical tag. Can I just say what the definition actually goes on to say, as it stands? It actually says that it includes, references to an embryo include an egg that's in the process of fertilization, or this, of course, you can see how this flows from Dollyland, is undergoing a process capable of resulting in an embryo. So one thing to note there is it talks about an egg in that included bit, which tends to knock out the idea of embryo models. But of course, the word including is inherently non-exclusive, which means they might be in there. So there's need for clarification. But as to this ultimate sort of circularity, perhaps the solution is to actually say that what it results in is something of ethical significance. So it could be that actually we say that it change the last word, it's capable of resulting in not an embryo, but say, by example, a fetus capable of surviving without intervention, birth. So there may be a way of marking like that. I find it a little bit circular. And of course, the effect is it just goes back to a court, and the court would then look around and go, what the hell do we do? And they'd go to the international stem cell case, which is the case I mentioned before. It's quite normal, I think, that we at the time in 2008, I remember everyone sort of really concerned about human animal hybrids. definitions, pah. You know.
Okay, let's move on now. It's another question for Julian in the first instance. And this is from Matt Jordan. It appears that reproductive organoids, such as those derived from stem cells, do not fall within the definition of an embryo in section 1 of the Act, as amended, blah, blah, and so are not regulated by the Act. This seems to be the case because fertilization doesn't take place and the capacity of organoids to develop into embryo / human life is scientifically uncertain. So then he says, is this satisfactory? And what are the relevant legal regulatory frameworks currently governing their use?
Okay, I think this is obviously going to be something to which colleagues will be able to speak very clearly. From a legal point of view, of course, it is true that an organoid is not an embryo. Just put it like that. However, the point you raised, Matt, I think is a really interesting one, an important one, which Sarah actually raised at the outset in talking about the fringy areas of if what can be done in vivo, for example. Now, essentially, if we're talking about a cell therapy, call it by its name, if we talk about a cell therapy, then as a lawyer, the sort of regime you look at is going to be one of tissue and cells, so quality and safety applies there. And also the regime akin to, wouldn't be exactly, but akin to what the advanced therapy medicinal products regulation. You have that in the UK. I say akin to because it isn't quite the same, because you're not marketing a product. But I think the framework there is relevant, and indeed the framework in relation to, say, genetic manipulation that we have in relation to gene therapy medicinal products is also relevant. We've got bits and pieces. Coordinating it in a coherent and responsive way is, I think, something of a job. I think others will have something to say on this.
Robin.
Yeah. I mean, an organoid is something we think of as very specific to a particular type of organ, but I think the thing that is of more concern in a way is the stem cell derived embryo models, not the so-called non-integrated ones, which clearly could not give rise to whole embryo. They're fine. But the integrated ones, where you have extraembryonic cell types as well as embryonic cell types, become a problem. Some of these are already fall foul of some regulations in other countries where you cannot even research them because they would be qualified as potential embryos. So that's an issue. But in the UK, they're not covered by the HFEA. If you're using an embryonic stem cell line, there is a review by the Stem Cell Bank Steering Committee, but one could argue whether that's robust enough. If they're derived from induced pluripotent stem cells, they're not even covered by that. So they're derived using induced pluripotent stem cells, they would effectively be embryo-like entities that are clones of the original donor of the somatic cell used to make the induced pluripotent stem cells. So the question becomes, should these be regulated by someone, and should they be regulated by the HFEA? The International Society for Stem Cell Research in the updated guidelines suggests that these should be subject to the same review and oversight as a normal embryo because they are very close. They could be very close to embryos. Now, at the moment, there's no evidence that they're close enough to give rise to an embryo if they were implanted, to give rise to a later embryo if they were implanted. But that sort of experiment is going to happen probably with non-human primate derived embryo models. And if it was possible to implant one of these in a non-human primate and get development to a much later stage and to a fetus and maybe even a lipor, you would then absolutely have to bring those under some regulatory control somehow to stop their use. So that's why I posed the question whether these should come under the any revised HFA Act. But they're not used to regulating this type of work at all. So that's going to be a challenge, perhaps.
The next question I'm going to take is from Alison Murdoch, who has asked two questions. I'm going to go with her second one first and perhaps use her first one as a summing up question at the end to you all. So she's said, immature gametes within the gonads are included in the scope of the HFE Act. Should this be removed from the legislation? If not, how would live transplant of gonadal tissue by transplant surgeons be regulated by the HFEA? What if the gonadal tissue to be transplanted contained derived gametes from stem cells?
Well, I mean, the HFEA doesn't, as far as I know, cover transplants of gonadal tissue at all. Someone has that must be covered by another regulatory body though.
So I'm going to let Julian come in because I can see he's agitating on my screen. So I'm going to go to him now.
Well, as Robin says, it isn't regulated by the HFEA. Oddly, it's not really something which is countenanced either by, if you like, the next big touchstone, which is the ATMP regulation. Forget Brexit, you probably want to anyway. But effectively, the ATMP regulation is the relevant regulation for somatic cells, products, human tissue engineered products, and gene therapy products, a special class of medicinal products. And for that reason, that sort of framework is sort of irrelevant because we're not marketing a medicinal product, but it is relevant in relation to quality. And I think if we look at, looking at that, one of the surprises to me is not only the fact that it never mentions the word autologous, which seems humorous and with hindsight, but also that it's completely blind to this issue, which for years has struck me as something that's standing out. It means it's just not simply not accommodating. But, and the reason I say that is because, looking at a definitional thing, you'd say, okay, somatic cell, did you just say somatic cell? So you've got them, or was that a gene therapy? It doesn't even fit neatly in those classifications. And actually that's where the courts take the sort of purposive approach that the House of Lords took in the Quintavalli case, and actually trying to work out, well, what is it meant? What is meant by this? And that's definitely true in relation to gene therapy and medicinal products, where there's sort of a pretense that even though edited products aren't really covered by the definition under the ATMP regulation, nevertheless, let's treat them the same way. And so they should, because actually the same issues, many of the same issues arise. There's some additional ones, there's some that don't arise. But you have to go with what you've got, and a regulator, I think, does have to take a view pending development of the law.
And what does the acronym ATNP stand for? Because I missed that.
I apologise. This is the Advanced Therapy Medicinal Products Regulation. You want the number? It's 1394 of 2007. And so it is a central plank in the regulation of regenerative medicine, but doesn't say regenerative medicine, and it is a wider brief than that. And today we look at it as applying to things like the use of CAR T cells and so on. So it is wider, but insofar as it actually encompasses a cell therapy, the grafting of tissues, that ought to apply if something falls in that framework. Of course, if it doesn't, it's just a cell graft, and it's a big subject in its own right. And I'd love to talk about it, but I think it would be a bit of a tangent. Suffice to say that the HFEA does have the ability under that act, under the HFE Act, to recommend changes in the law, and that is one which I think it could, helpfully consider bringing in. Whether it should be that regulator is I think, a separate question.
I think the HFEA, under the HFEA Act, I think they cover the uses of immature germ cells in vitro or to fertilize. They don't cover transplant into a person's gonad.
That's right. Yeah,
and that's the area which is I think uncertain, because
Yes, it's definitely not under that Act.
No.
I mean, should it be? Should we need the HFEA to step in whenever you want to go in and have a transplant of that nature? I don't know, but it is a question which I agree should be raised and be part of the public discourse.
I mean, it could be something that is regulated both by whatever the acronym is you said, whoever regulates that, and the HFA in conjunction as a combined thing, much as they do for or supposed to do for hybrid chimeras, for example.
Yeah,
animals containing human material. But
I mean, in the UK now that we're not in the EU, effectively it's the MHRA. So there probably is some room for discussion over that, as they have with, say, the HTA, as to Human Tissue Authority about where the overlaps are. It's not uncommon to do that, and of course they did with the stem cell bank years ago.
I'm just going to point out what someone said, but I don't think we'll go back to it. Is that anonymous attendee has said they're not sure that they agree that an ovary transplant would fall within the ATMP, but I think if we sort of keep going down this rabbit hole, we're getting a little bit distant.
Because it's an organ.
So yes,
possibly right.
Okay, so someone else has asked, how, if at all, does the development of artificial womb technology need to be considered in the revising of the definition of permitted embryos? Do we think it does, or do we think actually that the suggestion that was perhaps made before by something put in the reproductive tract, or I don't know how that would work with an artificial womb? I don't know quite what the plumbing is, so to speak.
In terms of advances in growing embryos outside the womb, it's approaching from both ends. So the problem of dealing with premature births is that there is progress there, and it's been animal work which is very encouraging, suggesting that you can use these sort of plastic bag-like things to keep embryos in a much more happy situation, or fetuses much more happy situation, so that they complete development properly. That's making small inroads, getting maybe a few weeks earlier. The other end, which is the one which would concern really the HFEA, of course, the growing of a pre-implantation embryo in culture up to 13 days and maybe beyond is relying on quite artificial 3D constructs. You could make those less artificial by using, for example, cells from the womb, endometrium, so that they will implant more normally, and that may be one way you can actually get these things to go further. But I think the chances of connecting the two, so you have ectogenesis entirely outside the body, is extremely remote at the moment, so I wouldn't be too concerned about that.
Would permitted use of sperm and eggs created from iPS cells provide those suffering premature infertility and same-sex couples the opportunity to generate eggs or sperm and have a genetically related children? If it may be possible in the future, what do the panel think about a change in the law?
Yes, I mean, clearly, if you could derive gametes, eggs or sperm from iPS cells from a patient who was infertile, then that would allow them to have, in theory, allow them to have children that were genetically related to them. They would those gametes would have gone through meiosis. It would be as in a normal situation. So it would be just like having sperm or eggs obtained currently in IVF. So that's no problem. To have same-sex couples have children, there's an issue there, because you cannot derive sperm from an XX individual, so chromosomally female individual. You need genes from the Y chromosome to make viable sperm. Having two X chromosomes is incompatible with making sperm. And the other way around, in humans, there's no evidence, or no good evidence, that you can have an egg, an oocyte derived from an XY, so chromosomally male individual. So you would have problems there in having same-sex couples having children, because you need eggs and sperm to make a viable embryo. Now, there are tricks that have been developed in mice which have allowed both ways around to happen, but they are so torturous and involve a lot of genetic manipulation, loss of chromosomes or whatever. These are really far too science fictiony to warrant even thought about at the moment.
Mary, do you agree with that position that Robin said? It's the science for that is just too difficult. As someone who knows about this difficult area of science using with embryos, do you think this is a bit too science fiction at the moment?
I think it's quite some way off, and also I think Robin sounded more optimistic than I am about getting gametes and normal meiosis happening from iPS cells in vitro.
I am an optimist generally, but I'm not the only one in this field. And if you look at the rate of progress from mouse to non-human primates, it's going very fast, and it just requires a few critical steps with the human material, and I think it will work. But I agree. I'm being an optimist, but it's not so far off. And if it happens, and when it happens, if it's in next year, 2 years, 3 years, you bet the demand is going to be there to use the technology to allow infertile people to have children.
Yeah, I think that we're good at making things that look like a bit like egg and sperm in vitro, but the evidence for having gone through meiosis is not so convincing.
Well, I think in the mouse and now in the non-human primate, it's very good. You get viable animals. So
with relatively low efficiency, though. But yeah, I think I would be very happy to see sort of genomic analysis to show that they've gone through recombination and we have exactly one copy of each chromosome. Mm-hmm. Is this sort of near to the Petri dish type analysis before you go for it?
For me, the biggest problem is less that. It's more the epigenetic changes that you may have inadvertently introduced by culturing things. And
we each have our pet concerns.
So I'm going to go now to the final question, which is I'm going to go back to Alison Murdoch and ask each of the panel in turn, what changes in the HFE Act related to the permitted embryo treatment and research would you like to see?
I think my overall thing with the Act is what Julian said about having this technology approval body, so that the Act doesn't have to be so specific. And that we could just have general principles in the Act, what is acceptable in law and what is not, and then sort of detail of what techniques are approved or what can be done would be sort of a downstream event by a technology approval body. I think this idea would make the whole thing a bit less fraught.
What Mary was just suggesting, and what perhaps is being suggested here, does in some ways go in the direction of the 2008 amendments, but which, as Robin pointed out in his initial contribution, was designed to simplify and allow generalization. And so one might imagine that being taken further. I suppose my note of caution would be that whatever one might think about, shall I say, some politicians, it's obviously crucial, and I hope we'd all agree that that such a change, which would be quite a major one to the sort of technology approval kind of happening downstream, that would need some really rather thorough public and parliamentary discussion before those powers were taken to such a body. So I think that would just be my caution there.
It's just about avoiding the scientific, the biological detail in the law and the technical detail, because that just, it's problematic, I think. And if it's agreed by society in principle, it's okay to use these assisted reproductive technologies, for example, to prevent transmission of mitochondrial DNA disease, then the law doesn't really need to tell us that, you know, you transfer a piece of a nuclear genome from egg A to egg B and you must remove it all. I think it just, in that particular case, it didn't leave enough scope for development. And it was at a pretty early stage of development. And, you know, we're always improving things.
Yes, I mean, it is often impossible to pick the social and the technical apart, but that does seem to be an example of something where you're really talking about very specific technical points that perhaps don't need such high level political input. Yeah.
Robin, what would you like to see changed very briefly, because we're up against the clock now?
I'm going to continue the same discussion, because I think it is sort of in a way nonsense to regulate the technique or the process, because they change very rapidly, and it's much, in my view, it's much more relevant to regulate the purpose. So in terms of mitochondrial replacement, if it had been to get, you know, an embryo without any abnormal mitochondrial DNA or very low levels of abnormal mitochondrial DNA, then it shouldn't matter whether it's polar body transfer, pronuclear transfer, spindle transfer, or whatever, or whether you use genome editing methods to eliminate the mitochondria carrying the mutation. The same is true for if you want to allow parents to have a genetically related children. The purpose of pre-implantation genetic testing is exactly the same as genome editing in that context. So it's solving the technical problems. Yes, that has to be done, and having acceptance of the use of a new technology is important, but the intention is exactly the same. So in terms of the question that was posed, I would quite like permitted, at least not necessarily immediately permitted, but I like the idea of having the sort of regulation making power, however you want to describe it, this sort of intention, which would make a subsequent change in the law much simpler than opening up the whole act again for in vitro derived gametes and perhaps for germline genome editing.
Julian, in the length of a tweet really now, because we've gone over time.
Okay, having a technology approval board is not unusual. I agree it should be a subject of fervent debate, but really that's what happens in relation to medicines regulation. So I would say that's not at all unusual. I'd like there to be a more responsive framework, and that is part of that same process. I think we need to work hard on the trust in the framework, and that includes a degree of transparency, which again, who recommended. And I think also, just finally, I would mention reinforce what Robin said actually about the 14-day rule, which is not directly relevant, but it's indirectly very relevant. And so I think certainly that is something which I would be up there with my list of my wish list of reviews.